Hi, everyone. Thanks for tuning in. My name is Jessica Smith. I'm the Director of Bioinformatics here at Sequel, and I'm also our moderator for today's discussion on transposase-based library technologies. Sequel was created to revolutionize NGS library prep to unlock the full potential of today's DNA sequencing instruments. And today's topic is critical to optimizing that potential. TN5 has become an integral component of NGS library prep. Its unique capabilities have unlocked a plethora of recent advancements now at the forefront of biomedical research and development. I'm joined today by two of my colleagues, Joe Meller, Sequel's co-founder and chief scientific officer, and Curtis Knox, our director of product management. Thank you both for being here.
Thanks, Jess.
Thanks, Jess.
Great. So let's get into today's presentation, which we've entitled Enabling Sequencing Applications with Improved Transposase-Based Solutions. Curtis, can you tell us a little bit more about what our audience can expect?
Sure. By the end of this discussion, we hope that our viewers will be able to first understand how tagmentation can work across a wide variety of NGS applications. Secondly, we want them to understand how transposase-based NGS library prep methods can result in significant workflow improvements. And then lastly, we think that they'll be able to understand how some custom TN5 loaded reagents can create opportunities for them for new innovations in their laboratory and be able to do all of this without compromising their data.
Thanks, Curtis. It sounds like there's a lot of ways that this enzyme can maximize the potential of NGS library prep. So I'm really looking forward to today's discussion. Joe, do you want to give us a little background on TN5 in NGS?
Sure, I'd be glad to. I think it's helpful for us to start with an overview, really, of where the sequencing arena is today. The growth of sequencers has become so prolific. We are now using instruments that are orders of magnitude higher throughput and larger than they were even a few years ago. And because of that, sequencing has really become a universal tool in life science research, not just for sequencing genomes and other samples, but really for a variety of types of measurement of biological assays. And I think it's important to point out that the cost and time associated with getting samples to those sequencers, which we call library prep, has really lagged behind and has not advanced and kept up really with the tremendous improvements in sequencer throughput. So Sequel's mission really is to focus on that bottleneck, how to create the tools that will enable researchers and users of sequencers to get their samples to sequencers more efficiently. And in many cases, that means we enable higher multiplexing of more samples and simpler workflows to allow samples to be processed more quickly and efficiently.
Wonderful. Thank you so much for that background. I think it really does sort of set the stage for this conversation. Do you want to get into a little more detail about the biology of TN5?
Sure. So, I think what you'll hear today really is that TN5 is a centerpiece of most of our products here at Sequel because it's an extremely versatile and a simple tool for helping scale a number of NGS workflows. Before we get into those products, though, I think it's helpful for us to maybe take a little bit of a step back and talk about the history of TN5. TN5 has been around for a few decades now, has been highly characterized, has a number of really interesting properties that owe to its natural capability of being part of a transposon. Transposons are mobile genetic elements in genomes, and this enzyme was evolved essentially the capability of allowing that DNA to transfer around inside a genome.
Thank you for giving us a little bit of history about TN5 and its biology. Now can you go into a little more depth about how TN5 is useful for NGS applications?
Sure. So I think there's a few really interesting properties about TN5 that make it a really versatile enzyme for NGS. I think probably the most important is that it being an enzyme that's capable of really binding and inserting DNA nearly randomly in any target sequence allows it to be sort of substituted for protocols that need a shotgun fragmentation-like function. Another really interesting property of the enzyme is that it can be functionalized, loaded with different types of payload, which we have turned into the ability to put different sequencing type functions into it, such as sequencing adapters. Finally, I think the other piece that we'll talk about today is that the enzyme is got a property that allows it to be exploited as a normalization agent because its function is to do its job and do it one time only, and therefore controlling the amount of enzyme can control the number of reactions that occur in a typical reaction.
Wow, well, this sounds like a system with a lot of potential. So Curtis, I'm going to ask you to tell us a little bit more about the various ways that we can use this technology in different NGS library prep applications.
Sure. So Joe talked about the biology of the enzyme itself, and what I want to talk about a little bit is how we've used that to create NGS library prep methods and products that really help the users when it comes to getting the most efficiency out of what they're trying to do. So what we've done is create a series of methods and products that allow for what we refer to as true multiplexing, so much higher levels of samples that can be multiplexed together for their sequencing. And we do this through a method in which we're utilizing asymmetric tagging of the libraries, adding the I7 section and the I5 section in sequential fashion, and that allows really nice control, allows normalization that Joe alluded to, and has high efficiency for converting the DNA into actual sequencing libraries. And so, as an example here, when we think about traditional ligation or other types of methods, you're typically looking at 5.5, 6 hours or more to do a plate of 96 samples. With our methods using TN5 transposase, we've gotten that down to right now 3.5 or less hours to do that full plate of samples, and at the same time creating some of those extra bonus features like the normalization that we talked to. And these have a wide range of applications, everything from whole genome sequencing, low pass genotyping, microbial and metagenomic sequencing, and even single cell RNA-seq from there.
Yeah, interesting. So you mentioned normalization as a bonus, but it actually seems pretty central to a lot of the products that we sell. Can you go into a little more depth about this unique capability and give us maybe an example of one of our products that leverages normalization?
Sure. So the first thing I'd like to maybe use as that example exactly is our PurePlex DNA library prep kit that we just released earlier in 2022. And this is one where we've taken those features and actually improved the workflow down to as little as 2.5 hours when it comes to that. And we've also incorporated unique dual indices into that particular kit. So that's something that we've heard from users that they're looking for extra control of their samples and data quality and everything else from there. So the incorporation of the UDIs has been very important as well. And that is a product that currently we have with 192 different UDIs. We plan to expand that up to about 384 in the near future as well.
Great. So you mentioned briefly that UDIs have something to do with data quality. Do you want to clarify that a little bit?
Sure. So when you think about making sure that the sequences that you get are definitively coming from each of your samples that you're interested in, because those samples all get pooled together on the sequencer, and it's very important that you be able to assign those reads exactly back to the sample of interest. Utilizing a unique dual indices allows you to make sure that you're fully separating that data out and have the highest confidence.
We've started talking a little bit about how our multiplexing capabilities can lead to improved performance. So Joe, do you want to give us an example about this and about how normalization works in practice?
Sure. So yeah, the idea of normalization is that in many sequencing workflows, the user or the researcher has their sample and maybe it's present in different amounts. And so it's a difficult problem, and labs spend a lot of time with this. And the way that our chemistry works is that it facilitates a sequential tagging process where the first tagging event is used as a way of specifying the number of molecules that can be generated. And then we pool that material together, or in the case of PurePlex, we have a different method for accomplishing this, to then turn those first tagging instances into a normalized library where all of those samples are represented with the same number of reads. So what you can see in this example, where we can take tenfold range of DNA amount and generate a consistent amount of library from that. This is a really useful thing for many people who otherwise would have to take those samples and either individually quant and adjust them, or make libraries and then normalize them afterward. The point is with our chemistries, this process is highly simplified, and we've removed for many people this need to do a separate normalization step.
Yeah, wow. Well, I can imagine a lot of different researchers and clinical applications where this would be really useful. So do you want to give us an example of how our multiplex workflows are used in the real world?
Sure. I think there's no better example in the last couple of years than labs that have had to scale up, for example, the ability to sequence COVID genomes. I think all of us woke up to a need really overnight, starting 2 years ago, where we wanted to be able to track and characterize different variants of SARS-CoV-2 genome that was emerging. And an example of this type of work was a collaboration we did with a group at TGen in Arizona, where essentially overnight we helped them stand up a COVID sequencing effort where they could take their COVID positive specimens and reflex them into a sequencing assay and track those variants in real time to better understand what was going on with the spread of the virus and new variants. And I think this is a great example where a lab was able to go from doing 0 of this type of sample to thousands per week in a matter of a few weeks.
That's a really great example of how the power of this kind of technology. Now let's look ahead at some new products we have. Curtis, do you want to tell us about what's coming down the pipeline?
Well, I think first, maybe, Joe, if you'd like to touch on some of the rapid, single-cell RNA-seq applications, and then, we can follow that up with some of the really exciting stuff that we've got coming down the pike here.
Yeah, absolutely. So, I think as we've sort of taken the Plexball and PurePlex technologies sort of as the basis and foundation for other types of assays that we can build, we look for applications where this need to multiplex or essentially process large numbers of samples is really important. And I think one really good example of this is in single-cell sequencing, especially single-cell RNA-seq. And again, here what we did is we collaborated with a researcher who specializes in this type of assay, was actually the inventor of a method called SmartSeq2 about 10 years ago, Simone Pacelli. And we created a kit that allows for this sort of integrated processing of single cells through to a sequencer-ready RNA-seq library in essentially a single day. And I think the story of this product is really that we were able to take the Plexball technology and combine it with a high-performance chemistry to essentially take RNA from cells and turn it into the DNA that becomes the input to our Plexball kit, which has, again, this really important property of normalizing for the differences that might exist between those samples. And so if you look at the performance of this product, this is where it really shines. Again, in a conventional sort of setting, the researcher would have this problem of having some cells generate more data than other cells, and they would have dropouts, for example. With Plexball, this problem is significantly improved because you get, again, greater performance across a greater number of samples and essentially more sequencing data from more cells in the same assay. And I guess the final piece of this is that we do this at really no ultimate cost to the actual sequencing performance itself. It's a workflow improvement. It certainly enables the multiplexing side of this. But if it produced worse data, this would not obviously be a great solution, we know, for scientists. And so we've benchmarked this, and we see that, we can have this really nice normalization without any real cost in terms of the metrics associated with detecting RNA from cells.
Great, wow. Well, I mean, between the COVID stuff and the single cell work that you just described, it seems like a lot of solutions are being generated by our company. I'd love to hear about what sorts of things we're going to help other researchers with.
Sure. So there's some really exciting stuff coming down the pike, including what we're referring to now as ExpressPlex, which is something that we have in early access at the moment and will be rolling out to users in the near future. And this is by far the fastest overall NGS library prep method out there to be capable of this true multiplexing and high levels of multiplex that we've talked about. And in this particular method, you literally are mixing your DNA, a tagging reagent, and a master mix you're putting those three things together, putting on the thermocycler for an hour, and then you're pooling and doing one cleanup, and in 90 minutes flat, you have 96 samples ready to go, all barcoded and ready to pool and put in your thermocycler. And we can continue that barcoding, multiplexing aspect upwards. We anticipate 768 barcodes in the near future, moving that upwards to 1536 and beyond as necessary from the users as we find that out. So we're really excited to bring this to customers in the very near future.
Wow, wonderful. Thank you so much for explaining a little bit about our technology ExpressPlex. But going back a little bit more to the transposase itself, are there any ways that we're leveraging that technology to do multiple different applications?
Sure. So that's a great segue into talking about something that we're referring to as Tagify, which is a TN5 transposase-based kind of toolbox, for lack of a better phrase, in which we are going to help users and researchers out there utilize the power of transposase-based methods and enable different types of innovations utilizing the enzyme along with some different tags to actually achieve new things. And that can be something like CRISPR verifications. It can be looking at variations where we're enabling ligation methods. We have unique molecular identifier methods and versions of this in which we're loading on sequences that will allow customers and users to really be able to hone in exactly down to almost a molecular level and look at the sequences that are coming through. It's a super exciting new thing that we're pushing into to help enable users to really create even more innovation in their laboratory.
Cool. Well, it's like really
Yeah,
wonderful
yeah.
that we have this really flexible transposase-based offering. I wonder, Joe, if you have a little bit more to say about sort of the application areas and the technical details about what we can do to help our customers.
Yeah, absolutely. I think as Curtis mentioned,
Yeah.
we're working in this Tagify arena. It's really about creating tools that, again, our users and those researchers can take and plug into assays that they're trying to build. Again, we realize we have some great products that help in multiplexing, but again, some of these are also just basic tools that can help build the next generation of multiplexing tools. And one great example of this is again, the CRISPR QC methods, which benefit often from being able to put a sort of molecular counter into the reagent that's being used to generate a library. And again, there's other ways of doing this. Again, the tried and true way that this field has used for many years is to do a fragmentation and ligation-based assay. And again, this is a great example where a transposase can come in and take what was previously several distinct steps in a workflow and replace it with a single step that has equally high performance and again, simpler workflow and more predictable performance. So I think we're really, again, excited to work with people in this field. We see that, again, for this field to mature, there's going to need to be more tools, and we're really excited to have the opportunity to be involved in building those tools.
You mentioned more tools, and I should be clear that most of the work that we've talked about so far has been on Illumina platforms. Everyone knows Illumina. But we also know that there are more tools coming into the sequencer space too. So do we have any plans to integrate our library prep with those other sequencers?
Absolutely.
Yeah, definitely. This is a really exciting area. I mean, I think the history of NGS till this point has really been the story of one very dominant sequencing platform, which has really built this market. But we now see, again, and I think it's exciting, the emergence of other sequencing platforms with, again, slightly different chemistry, slightly different abilities that are going to give researchers and NGS users more choice in what types of platform, but also, really specifically tuned perhaps to certain assays that they want to run. I think we've been very excited to start to work with some of these newer platforms. And for example, we collaborated with Sean Levy and his team at Element Bioscience to validate our Plexwell technology on their new Aviti sequencer. And again, we're very excited to see this technology, and we see that it certainly has a couple of benefits over existing sequencing platforms in terms of its flexibility and cost, and some metrics that are important, such as duplication rates and so on. As these technologies progress and other sequencers are made available, we're certainly, I think, going to be in a good position to use some of the same library prep technologies that we've built into our kits. And we think multiplexing will be an important part of all these platforms. We know that, again, users and scientists simply want to be able to run more samples and do so more reliably. And so we're very excited about that.
Oh, wonderful. I mean, I helped out a little bit with some of the element work, and I have to say the metrics that came off were really impressive. I thought it was really interesting to be working on. All right, guys, I'm going to let you both say at least one more thing about transposase and library prep if you'd like. So, Joe, I'll let you go first.
Yeah, I think, again, for us, as you've probably learned today, and again, hopefully our audience has learned, the TN5 transposase is a real centerpiece for our work in building better NGS workflows. And we think it has some intrinsic benefits that, for example, enable more scalable performance, and again, inherently sort of address some of the core issues that users face every day, like how to process more samples, save time and money and effort associated with things like normalization. And I think what you're going to see from Sequel as time goes on is, again, we're going to be taking some of the same approaches we've taken to date in terms of taking workflows that would otherwise be more complicated or take more time for a user and build workflows that essentially remove steps or simplify steps, for example, with new formulations of these enzymes, combining them with other enzymes as we learned about today with ExpressPlex. Again, very exciting product where we're able to essentially do many things in one reaction mixture. I think this is going to be a familiar theme in sort of how we engineer workflows to do the next generation of assays. And certainly as far as the TN5 enzyme itself, which is really the star of our show, I think we're very excited on all of the improvements that we see that lie ahead in terms of what we can do with that enzyme.
Yeah, and I would just add to that, hopefully people have seen and can maybe understand that we're really still scratching the surface. There's still a lot of improvements, a lot of advancements that we're continuing to make using TN5 transposase as the base module for that. And it's not just about what we produce and put in a box. It's really about helping our researchers and our collaborators to make new innovations and to do great science. So we'd encourage anybody who's interested in learning more about this or wants to talk about a project and say, what if? Reach out to us. We'd love to have that conversation with you and see if we can't help.
Great. Well, thank you both for today's informative presentation.
Thanks, Jess. It's been great.
Yeah, thanks, Jess. Really appreciate it.
And another huge thank you to our audience for tuning in. Feel free to reach out and let us know what you found most interesting about this discussion. We would love to hear from you. You can also connect with Sequel online at sequel.com to find additional resources on this topic and others. And of course, don't forget to follow us on your favorite social media platform. Once again, I'm Jessica Smith, Director of Bioinformatics at Sequel, and this has been Enabling Sequencing Applications with Improved Transposase-Based Solutions.